Well, good morning. Thank you all for being here. My name is Andy Bredemeyer. I'm Senior Vice President at the Medical Center of Excellence at Velsera. And today I'm looking forward to telling you about our platform, the Clinical Genomics Workspace, for clinical NGS test workflow support and reporting support. I'll focus on hereditary cancer testing as an application, but we won't limit our discussion to that, and I invite you to ask questions about the full range of what CGW can do. My objective today is for any of you clinical laboratorians here in the audience to help you consider how Velsera can assist you with helping stand up a test to validate one or more clinical NGS tests, and for those tests to develop a repeatable, efficient workflow for reporting. So let's get started. For those of you who may not know Velsera, you may have heard of one of the three entities that came together to form Velsera last year. These are three companies that are really well-suited. They have very complementary capabilities. So Purion, you may have heard of, the company I've worked at for 8 or 9 years, and its Clinical Genomics Workspace for secondary and tertiary analysis for clinical NGS and reporting support, and our clinical services that I'll speak about some more today to enable labs to get started, help them with their workflow, including variant analysis and sign-out where helpful. The Seven Bridges platform is an analytics platform for running secondary analysis type tools. There's a collection of over 800 tools that have been integrated on that platform, and you can build workflows and pipelines to perform analysis from NGS-based and all manner of other bioinformatic analyses and statistical analyses. So our customers on that platform have done a lot of research, including clinical and translational, but now it's also integrated with CGW to enable these workflows to be used in a clinical reporting workflow where appropriate. Those can be validated for clinical use. And then FastFinder is a platform from the company Ugentech, which is also now part of Velsera, that is for wet lab orchestration. So tying together multiple wet lab instruments used in a wet lab workflow, collecting QC data, and establishing provenance of samples and their QC data digitally to move away from paper-based and spreadsheets all over the place, but one unified system. And we've built that was built for PCR initially and was very popular for use for COVID testing and remains so. And we're building this out for NGS wet lab workflow as well. So Velsera overall has the mission of tying together clinical and research omics to drive clinical discovery more quickly into research discovery more quickly into clinical use, clinical testing, and then leverage the real world data from clinical testing back in the research realm. Velsera has a long history of supporting clinical NGS assays across a wide range of sequencing technologies, assay vendors, and indications. Over the past 10 years, we've had run on our platform over 200,000 clinical cases across our customers' laboratory instances. This represents dozens of different LDT assays, wet lab assays that we've integrated into our system, and that has turned into over 500 different orderable procedures, including some different gene subpanels from individual wet lab assays across nearly 200 customer laboratories. And among those are some 200 orderable procedures for hereditary cancer and other inherited disease indications. And you can see some examples there of types of indications that our customers have run on our platform. Hereditary cancer testing, which we'll focus on today, we've supported through several commercially available assays from Thermo previously and Illumina's TrueSight hereditary cancer, which you may know and we'll talk more about today, as well as some custom LDTs that laboratories have developed over time and we have implemented on our platform. So what is this Clinical Genomics Workspace, the CGW? This is an end-to-end tool for supporting clinical NGS from accessioning and the fast queues off of the instrument all the way through secondary, tertiary analysis, interpretation and reporting, and then returning that result to the electronic health record. So some core capabilities here are first, you see on the left, prioritizing variants based on their clinical significance. So using our curated knowledge base, publicly available data sources, databases that we can use, we integrate, and then have our software logic operate on those pieces of data to automatically place variants across the ACMG classification scheme. Or in the case of somatic cancer, which we also support, the AMP-ASCO-CAP scheme for variant classification. We also have a database, a library of clinical interpretations that users of our platform across many customer laboratories have written and put onto reports and signed out for patient cases and returned to the medical record. And those you have access to, de-identified as sort of opinions of your peers about how a variant ought to be classified and what ought to be said about it from a narrative perspective in the interpretation. So that's also a key feature that our customers really appreciate about our network and our platform. And then, of course, when this work is done, the professional judgment has been applied. You need to put these results onto a concise report that the physician and their patient can understand, and gets them the information they need to manage the patient. And so we have lots of options for how to report. We can configure those in many different ways and apply your necessary branding and whatever you need for your customers' needs for reporting. And then I don't want to neglect that we also have done a lot of integration up and downstream with LISs, with sequencers, in order to get cases automatically accessioned from your LIS into CGW to get sequence data in for secondary analysis, and on the other end to be able to push back the results, be they PDFs, text files, or structured JSON format reports to the electronic health record. All right, so we found in the past few years that hereditary cancer testing is really well supported by Illumina's TrueSight hereditary cancer test. This is the most compelling commercial offering that's out there right now for this type of testing in a targeted panel fashion. And so this is already well integrated for several years in CGW. It's ready to go. If you want to run this test, we have to configure your report to be the way you want and set, say, some variant filtering thresholds based on your validated criteria, your performance characteristics for your assay. But otherwise, it's ready to go. So how does this workflow work on CGW? Well, secondary analysis is performed today with Illumina's Dragon enrichment pipeline, and you provide those files into CGW. And then we perform several analyses. We automate the quality metric review to ensure that you've got good quality data from your sequenced sample. And then you can move to looking at individual variants and assessing the quality. And we'll spend some more time on this in detail. Once you've identified which variants are real, you move to the classification, interpretation, and reporting, where the CGW has automatically, based on its knowledge base, triaged variants into the various classifications across the ACMG scheme, and then you can go into our ACMG guided workflow, review the evidence codes that have been automatically assigned to determined, and then you can review the evidence and make your own determinations about how to change some of those evidence codes and recalculate the classification automatically. Then you can leverage our interpretation library in order to have some assistance with how you write up that interpretation. So our customers have reported that these interpretations help them gut check their opinion about how to classify and describe a variant against their peers, other experts at other laboratories. And then finally, we render this into a PDF report. You see an example here of how your report could look. You see a clear indication of whether it's positive for a pathogenic variant or not. Some very brief details, and then the more detailed interpretation below, as well as you can see some, maybe you can't read, but coding exon coverage data to indicate which exons and which genes may not have had sufficient coverage, which is an outcome of the variant or the sequencing quality review that you'll have done earlier. But you can communicate that easily to your physician customer, so they know where you may not have had the best coverage and may not be able to confidently say that you have been able to detect variants. I want to point out that, as I made mention of, one assay can be configured to be multiple different orderable procedures in CGW. So if you, for instance, want to report all 100 and however many genes there are in true-site hereditary cancer, you can do that. But if instead, for either reimbursement reasons or because of what your physicians want to order, you want a smaller subset of several genes, or maybe a specific cancer subtypes you're looking at trying to detect a hereditary predisposition for, you can do that. I know that some insurers pay for BRCA1 and 2 and not the whole laundry list of other genes, and you can configure a panel, orderable panel, to just report those 2 genes and have that option for those physicians or patients where that's the request. So that's easily done, and is a way to forge a path towards reimbursability. All right, no, lest you think that we are an Illumina-only shop or a true-site hereditary cancer-only solution, that's not the case. We have experience with lots of different assay vendors. We have integrated assays from Thermo, from Pillar, Archer, PGGX, and others, as well as a number of custom solutions, LDTs, bespoke LDTs that our customers have built in their lab and developed their own secondary pipeline or developed it with us. So all of these things are supported in our system. I also want to call out that, especially with the integration of the 7 Bridges analytics platform with CGW, we now have additional power to employ, for instance, Velsera's own pipelines, like our whole exome or whole genome alignment and variant calling capabilities, that then you can validate and integrate into, we've integrated into CGW so that you can report out disease subsets perhaps using those technologies as the backbone. Or if you have your own pipeline that you want to build or you want us to integrate into the platform, we can do that. So it's a lot more flexibility here for custom secondary analysis pipelines and really robust performance of those on a repeatable basis in a validated fashion within our system. Otherwise, the workflow remains as described in the previous slide for these new solutions for integrating other assays or plugging in new secondary analysis capabilities. So let's look in more detail at quality review as a major step in the workflow. We provide, based on the BAM file, a view of the basic read and coverage metrics shown on the left. So how many reads did you get? What % are on target? What % of the target space has achieved some depth of coverage that you want to achieve, depending on your application? 50, 100, 250, or if it's somatic cancer, maybe 1000x. You can see on the left here that you can set a sort of a threshold or an average from your validation data, and then if that threshold is not met, we'll highlight for you that, hey, this metric is below your spec, and this is an example of a pretty lousy sample that didn't get enough coverage, and you see a lot of yellow there because the performance wasn't great. And then on the right, you can see, as I alluded to before, exon coverage metrics. So in which exons of which genes did you fail to achieve coverage of, let's say, 50x across some percentage of the gene, of the exon rather. And those then, as I mentioned, you can report on the report itself. And then likewise for clinically actionable positions in the genome where there's something known and that there's reason to make sure you got good coverage, you can view those as well and disclaim as necessary or repeat the sequencing if you can do better based on your assay's performance. So now once you've demonstrated that you have good quality sequence data for that sample, let's look at the variants and determine what here needs some scrutiny in terms of what's a real call and what's not. So we take the philosophy that you should be able to set, based on your validated filter criteria, some threshold that if you exceed that, there's no real reason to review in detail the quality. And then some that completely fail, that are very poor, you also don't need to try to rescue because they're just not good enough. But somewhere in between, you may have some gray area where you need to look. You may need to open IGV. You may need to determine whether there's a sequence context concern or something that gives you pause for reporting a variant. So you may be able to see that in this screenshot we see we're at the quality review step, and there's several buckets that we have automatically triaged variants into, whether they've automatically pass or fail, or they're somewhere in between that need manual review. And then based on variant type, you can select different tabs, and you can see visually see the sort of graph-based filter that's applied. In this case, there's a variant allele fraction, a depth filter for small variants, SNVs, MNVs, indels, insertions, deletions. And you can step through each variant type and see each variant type's own filter criteria, and then see the variants in the table that meet those criteria and perform a review. And then you can always launch IGV directly, and that takes you directly to the spot. You can see the read pileups from the BAM. You can see the VCF call there on the top track. So this is a way to examine any sequence context issues or strand bias or all these sorts of things that you might need to examine in IGV before you determine whether that call is real. And then you indicate that on the system that this passes review, and you can move on to interpret. All right, so identifying clinically important alterations is automated in the system based on the knowledge base, both our curated content and the databases that we have loaded and the interpretations from our network of users. And so that you can then review manually and make your decisions about whether you agree or want to, based on other evidence, change the assignment of certain of the ACMG codes, and then finalize your ACMG classification in this guided workflow. You can leverage that library of interpretations that we have to get leads on additional publications you might want to review, can automatically drive classification if there's an opinion out there from one of your peers that something should be in a given classification level. And then, as I mentioned, you can use those interpretations as a starting point to write your own narrative interpretation, or even use wholesale what someone else has written if you like it that much. All right, now beyond the capabilities we have in our system to automate your workflow, we have a really strong team of scientists, medical professionals who have either been in labs themselves and have done a lot of this work, or have worked with many of our customers over the time to perform validations to other services. And so I think this is a really a claim we can make as Velsera, that we're very strong at enabling labs who may be just getting started or may have certain competencies they're still bringing up to speed, where we can help fill that in. So one is validation support, where we can help design your entire validation strategy, help you acquire samples, plan out your runs and how to be efficient about how you run your various aspects, different parts of your validation, and then perform the analyses and even draft a validation report for you for your review and editing. So any mixing and matching of those parts are available if that can help you get started. We have a whole suite of lab launch services, which includes gap analysis, mock audits, even writing SOPs for you and training your staff if you need it. We have folks who are really skilled and experienced at that. Revenue cycle management services refers to getting paid, right? Getting reimbursed for your test. We can help you determine whether, given your payer mix and the patients that you're going to serve, you may need to seek MolDX approval, or how you should go about ensuring that when you submit for reimbursement, your test will be reimbursed. And then finally, our interpretation services and sign-out services. We have a team of variant analysts who can pick up after the automated report generation that is done by CGW and do the first pass of variant assessment with respect to quality and clinical relevance, and pass it back to your team for final review and sign-out. We can even perform sign-out. Our boarded medical professionals are capable of doing that for you as well. And I'll speak more about some examples where we have one of our customers has leveraged these services. And that example is Solaris Health Partners. This is a large network of urology practices in the United States. Over 600 providers in 13 states. And they had a pathology laboratory and did molecular testing, PCR, but they saw a need for offering hereditary cancer testing to their urology patients. So they diagnosed cancers in these practices, and they're not oncologists, but then they recognized that patients really wanted quick access to understand whether their prostate cancer or other urologic cancers might have a hereditary component, and so they wanted to be able to offer this testing. And we picked it up from them as they were kind of struggling to get validated, and we were able to help them validate the test and launch. So their first lab that worked with us was Integrated Molecular Professionals, a New York-based lab. You can see some of their great folks there whom we've worked with. So we were able to get them stood up and launched, and they've been live for a couple of years. Then Solaris said there's a need elsewhere at our other practices, and so with a handful of other labs, we are also assisting them with getting set up in CGW, as well as providing these sort of wraparound services to help, including examples here of helping them create a business plan and plan for reimbursement. Even determining what's the instrumentation that a lab needs in order to stand up a certain assay that we've jointly decided might be helpful for their physician customers and their patients. Drafting SOPs and training staff, as I mentioned, validation support, planning, orchestrating, and performing the analyses, implementing in CGW, of course, to be able to run the test, and strategizing for and preparing for MolDX submission, as well as the interpretation services and sign out. So through this set of services and using our platform for their hereditary cancer testing support, Solaris has is working on standing up 4 more labs, and they're able to bring this type of testing to a lot of patients and their families who sought it. And I think this is a good case study, an example of how Velsera can help a large network of several labs that are all at different, in different parts of the country, have different geographical considerations, including payer mix and different capabilities. So we kind of met them where they are, and we're able to supplement with services that they needed for each site. And so should your lab need any of these types of services and support, we're very interested in finding out how we can help you do that. So I guess I'll close with, indicating that, we're ready to help in many of these ways I've described. We have, we're building workflow orchestration for the wet lab, and that will be coming soon. But we're very strong on secondary analysis today, both sort of already set up pipelines that are appropriate for assays you can buy off on the market today, or your own, should you have one or want to build one with us. And with respect to variant quality assessment and interpretation, classification, and reporting of variants, that's our bread and butter we've been doing for a decade. Again, the services that you may need to get your test validated, get reimbursed, and if you need help with bandwidth for interpretation and sign out, we've got you there too. So I'd invite you to visit us at our booth 1211. You can also scan this QR code if you want. We have a 2-page document that gives some useful information about if you're trying to stand up a hereditary cancer test, what some considerations are and how we can support you. Feel free to download that. And with that, I have maybe 4, 5, 6 minutes. I'll take any questions you have. Thanks for your attention. The question is about copy number variants and if they can be interpreted by the system. Yes. I think the example I showed there might have been from a somatic cancer test. So you saw fusion, splice variants, copy changes, and small variants. But yes, all those variant types are, can be ingested from variant calls from a Illumina or other vendor pipeline, or we can make those calls right from FastQ if the proper data are available from the assay. And yes, and then we can automatically interpret like we can any other variant type. Yep. I mean, assuming that there's something to be said about it from a clinical perspective. Yep. I'm happy to take any questions in person up here at the end as well. Thank you.